So, hi, welcome everyone. We're continuing our series on cancer. Today we're going to look at cancer biology. And I think it's interesting when we look at epidemiology of cancer, the incidence of males, females, and children differ, but predominantly for males, the cancer incidence is greatest for number one, the prostate, second being lung, third being colon and rectum. For females, cancer incidence is first in the breast, second in the lung, and third in colon rectal. But mortality differs. For the male and female, uh, death from cancer of the lung is the cancer that usually takes lives, whereas prostate and breast come second, and colon and rectal come third. In children, the incidence of cancer greatest is leukemia. Second is the CNS, and third are neuroblastomas. And in mortality, leukemia is usually most fatal. Second is brain tumors, and third is neuroblastoma. So those are some of the epidemiological factors associated uh, with cancer. So in cancer biology, we take a look. A tumor can be benign, pre-malignant, or malignant, whereas cancer by definition means malignant or it spreads. So we know that cancer is a group of diseases. It's characterized by unregulated cell growth and the invasion and spread of cells from the site of origin or primary site to another site of the body. When we look at cancer, we have our oncogenes and our tumor suppressor genes, and we have apoptosis, and these are important in defining characteristics that actually can help us. We have a set of uh, genes and signals in place to prevent cancer and unregulated growth of cells from occurring. So when we look at cancer, here's our normal cell. It can be uh, no cancer, where everything is functioning, or we could lack a tumor suppressor gene, or an oncogene can be turned on. We want apoptosis. Apoptosis is basically the body's way of killing cells we don't need. It's important. It's not a bad thing. We need apoptosis. For just normal cellular regulation. It's kind of our waste system. Excuse me. So we take a look at oncogenes, uh, and we have what we call tumor suppressor genes, like your retinoblastoma and your TP53. When retinoblastoma or TP53 is turned off, because they're the regulators, they're the gatekeepers for cellular growth, and they're really important because they're the ones that tell when to make cells and when to not to make cells. And when you take the boss off the line, uh, nobody controls the line. That means there's no brake, right? No brake, assuming that the car is moving. There's no brake, it's going to keep moving. Cells are going to keep replicating. So the active oncogene gets turned on. That's when this happens. No breaks, no breaks, and then we just keep getting replication of unneeded cells. So an oncogene is a gene that has the potential to cause cancer. You might be thinking, well, no, I don't have any of those. Oh yeah, you do. We all do. They're in our body. Um, and most of the time, thank God, they're not really active. According to Knudsen's two-hit hypothesis, they become active once you get a second hit. So if you have an instability in a particular gene and then something happens with ionizing radiation. We're not talking the Hulk here, okay? We're talking, something disturbs the DNA. It alters the genetic code. It alters the gene. Then what can happen is that's the second hit. The second hit means, well, game over. We're going to start replicating now. We're going to have aberrant cells going crazy. A proto-oncogene are normal genes involved in the regulation of controlled cell growth, like your TP53, your retinoblastoma gene. When the proto-oncogene becomes mutated or overregulated, it's called an oncogene, and it results in the unregulated cell growth and transformation. So proto-oncogenes in normal cell growth. So you look at the growth factors here. Here's your nucleus. Everything takes place in the nucleus, right? It's the master cell. So, what happens in this DNA? What becomes mutated? Is it the transcription factor that causes a mutation? Is it a signaling mechanism like a kinase or a cyclin? Is it a receptor mutation? Is it a growth factor mutation? They're all different. All these circles are different mutations that can cause eventually cell proliferation. So the proto-oncogenes encode proteins that function as growth factors, growth factor receptors, signal transducing proteins, and nuclear transcription factors. So growth factors, okay, can have a mutated growth factor. Now you would think that if a mutated growth factor is mutated, it wouldn't be able to bind with its receptor. But here's the thing. Earlier stages, it still can. And then once it mutates, it doesn't need to do that anymore. Let's look at the cell cycle. And G0 is what? The quiescent phase. Everyone's just chilling, sitting at a coffee shop, just drinking coffee, waiting for their name to be called, kind of like firemen, waiting, for when they have to, run. The aberrant production of proteins can result in an aberrant transition from G0 quiescent to G1, which is your replicative phase, where the chromatin starts to the nucleolus starts to degrade, and the chromatin starts to come out, and it starts to form till we get to the S phase. And this can cause subsequent growth. Growth factors. Fibroblast growth factor are a family of proteins that are normally expressed during the proliferation of cells required for normal wound healing, but overexpression can lead to tumor formation. To give you an example, keloids. They're not necessarily tumors, but they are, if you've ever seen a keloid on an individual where a scar doesn't heal, I mean, it heals, but it just keeps proliferating until you have this mass where they had injured themselves. A lot of this has to do with the fibroblast growth factor. Platelet-derived growth factors are polypeptides that are important for the extracellular matrix production, but overexpression can result in a proliferation through autocrine stimulation. It's just automatic. That's what autocrine means. Endocrine means it needs stimulation through a different site. So growth factors can mutate, and once they mutate, what do? They send a mutated signal all the way down to the master switch. It's like aberrant electricity, if you will. So the structure of cell surface receptors. I want you to think about something for a second. If a tumor cell is located in a capsule within the tissue itself, and it's encapsulated, but it's anaplastic and has multiple mitoses, and it's lost it's becoming, anaplasia. There's significant anaplasia going on. If that's not taken care of and a body can't rectify it, it can pop through the basement membrane. And once it does that, it can metastasize. So you have proteins called E-cadherins at the um, at the basement membrane that are eventually eroded by metalloproteinases and collagenases by the tumor itself to separate the cell-to-cell adhesion, and it's able to spread into the lymphatic and hematogenous system. But here's a transmembrane protein. This is your lipid, your bilipid membrane, right? You have your lipids on the outside, your hydrophobic lipids, and then you had your hydrophilic inside. You have membrane channels. This is your extracellular domain that has your glycoproteins and receptors scattered throughout it, as well as your G protein coupled receptors, your G alpha S, your G alpha Q, your G alpha I. Here's your transmembrane protein and your intracellular domain. The intracellular domain is your cytoplasm of the cell. And this is where the receptor interacts with the interior of the cell or organelle, relaying the signal. And a lot of this is done with tyrosine kinase, which is a protein. And it's a phosphorylating protein. And we'll see throughout biology that phosphorylation has a lot to do with turning off and turning on different mechanisms in the body. So growth factor receptors, growth factor, here's your epidermal growth factor receptor. And this is rearranged during transinfection, which is your RET proto-oncogene, which is implicated in multiple endocrine neoplasia type 2, which can cause medullary thyroid cancer, pheochromocytoma, marfanoid habitus, and type B is your medullary thyroid cancer. It's your pheochromocytoma, and I believe it's your hypoparathyroidism. But these are mutated growth factors. Overexpression of the growth factor receptor known as HER2/neu is associated with the development of breast cancers. So it's in the body, right? Mutations in RET are commonly associated with multiple endocrine neoplasia, or the MEN syndrome, including MEN1 and MEN2. Actually, MEN1 is MEN1, MEN2, A and B are RET. The next level, which detects and the cell growth and development can occur at the level of downstream signaling. Two such examples are the RAS proto-oncogene and the non-receptor tyrosine kinase proteins. These are all proto-oncogenes that basically, once their brakes are removed, they just go crazy. So here's your growth factor. Here's your receptor, your transmembrane protein monomers, tyrosine kinase. Here's your adapter protein. Here's your guanine nucleotide exchange protein. Guanine is part of your DNA. Here's your GDP, not your gross national product, but guanosine diphosphate. Here's your RAS protein. Here's GTP, guanosine triphosphate. And then what happens is you have a protein kinase cascade, phosphorylation, transcription factors get activated in the nucleus, and then we have gene expression. So the RAS is most commonly mutated oncogene with cancers in 10 to 20% of tumors harboring RAS proteins, and they're found in a large number of tumors such as colon, pancreas, and thyroid. Nuclear transcription factors, proteins such as your proto-oncogene MYC. Now the MYC is implicated in c-MYC is Burkitt's lymphoma. It's very aggressive. It's mostly found in Africa. And then you have NMYC for the neuroblastoma. It integrates divergent growth promoting pathways and ultimately leads to the production of proteins that allow the cell to advance through the cell cycle rather than stop. So they basically are able to shut down the p53 and the retinoblastoma chain. Nuclear transcription factor WNT, or WT1, is encoded for chromosome 11, and it's mutated in Wilms tumors. It's a common kidney tumor in children. So now we look at tumor suppressor genes. There's many, many more proto-oncogenes that once released, there's no system to break them. They will continue in vigorous proliferation. So I give you some of those receptor tyrosine kinases and non-receptor tyrosine kinases. We have the ALK, that's the name of the gene, just ALK, it causes lung adenocarcinoma. We have the endothelial growth factor, or ERBB1, which is a receptor tyrosine kinase, can also cause lung adenocarcinoma. We have the HER2 that you just saw, or ERBB2. It's also a receptor tyrosine kinase for breast and gastric carcinomas. We just looked at RET. It's the receptor tyrosine kinase. It causes MEN2A and 2B. Medullary and papillary thyroid cancers and pheochromocytomas, which are tumors of the adrenal gland. We have BCR and ABL. These are non-receptor tyrosine kinases implicated in chronic myelogenous leukemia and acute lymphocytic leukemia. JAK2 is a proto-oncogene. It's a non-receptor tyrosine kinase. It's implicated in myeloproliferative neoplasms. We have BRAF, BRAFF. It's a serine threonine kinase implicated in melanoma, non-Hodgkin's lymphoma carcinoma, and papillary thyroid cell leukemia. Papillary thyroid cancers and hairy cell leukemia. c-KIT, C-KIT, or cytokine receptor CD117, is implicated in gastrointestinal stromal tumors, as well as mastocytosis. c-MYC, what you just saw, is implicated, as I mentioned, in Burkitt's lymphoma. MYC-N, or NMYC, is a transcription factor implicated in neuroblastoma. KRAS, or RAS, is a guanosine triphosphatase implicated in pancreatic, colorectal, lung cancer, and endometrial cancers. And BCL2 is an anti-apoptotic molecule that inhibits apoptosis, and it's found in follicular and diffuse B-cell lymphomas. And we talk, apoptosis is basically your body's way of destroying and eradicating old cells that are no longer needed, and they're broke down, starting at the nucleus through pycnosis, where the nucleus starts to break down, and you have karyohexis, where the nucleus starts to degrade completely and the membrane becomes loose, and then karyolysis, where the whole cell just kind of lyses. And then, of course, your body comes and phagocytizes the cell and its components. Tumor suppressor genes, okay, we're going to look at that. Tumor suppressor genes, they basically suppress the proliferation of cells. So they downregulate. Your upregulators are more of your pro-oncogenes, where your tumor suppressor genes are downregulators. And some of those, they lose their function, right? So oncogenes gain a function, proto-oncogenes lose their function. Those are your p53 and retinoblastoma some of the big ones, the gatekeepers of the cell genome. Loss of function basically increases cancer risk, and this is where you have both two alleles of a tumor suppressor gene, must both of them express the disease. This is what Knudsen's two-hit hypothesis means. So some of these tumor suppressor genes, and these are the ones that we want them to work. We really want them to work. APC is a negative regulator of beta-catenin and the Wnt pathway, WNT. When these are no longer expressed or they lose their function, it can cause colorectal cancer. And the WNT or Wnt pathway is associated with familial adenomatous polyposis. And how do I remember APC and beta-catenin and the WNT pathway? I think as a APC, like an MPC in the Army, bet he didn't go to the canteen but went that way, or he went to the canteen. Um, you got to think about ways of memorizing these things. BRCA1 and BRCA2, we call them BRCA. These proteins, BRCA1 and 2, help prevent breast cancer, ovarian, prostate, and pancreatic cancers. So BR for breast, CA for cancers. CDKN2A is found on the small arm of chromosome 16, and it blocks the G1 to S phase, right? S is the DNA phase where DNA is being, the new DNA is being produced. When that phase is, not blocked, so for instance, when you have a loss of function of the CDKN2A, then the DNA can't be checked. So obviously this can cause many cancers, such as melanoma, lung, and pancreatic. DCC, tumor suppressor gene, it's deleted in colorectal cancer. SAMED, or DPC4, is deleted in pancreatic cancer. So basically it means when it's no longer, when it loses its function, it can cause pancreatic and colorectal. MEN, the multiple endocrine neoplasia 1, or the MENIN gene, is responsible for multiple endocrine neoplasia type 1. NF1 obviously is the neurofibromatosis type 1. Its gene product is neurofibromin. It uses the RAS GTPase activating protein. Neurofibromatosis type 2 or NF type 2 uses the Merlin or schwannomin protein. The P10 is a negative regulator of PI3K, AKT pathway for prostate, breast, and endometrial. RB1 inhibits, or retinoblastoma 1 inhibits the elongation factor 2. It blocks also the G1 to S phase. That's a good thing. We want the checkpoint. Unblocked, unregulated, loss of function will result in retinoblastoma and osteosarcoma or bone cancer. And TP53 activates P21. It blocks the G1 to S phase. You can see how important the G1 to S phase is. And most cancers are associated with this, such as the Li-Fraumeni syndrome, which causes multiple malignancies at a very early age, such as sarcoma, breast, brain, lung, leukemia, and adrenal cancers. And that's pretty massive. I've actually met someone who had a family history of that. The TSC1 is your tuberous sclerosis complex. It uses the Hamerton protein. The TSC2 or tuberin 2 causes this also tuberous sclerosis complex. VHL is your von Hippel-Lindau. This is also responsible for a lot of your breast and endometrial and kidney. It inhibits the hypoxia inducible factor 1 alpha. The WT1, not to be confused with WNT, is that mutation is responsible, well, the gene is responsible for the urogenital development transcription factor and is found in Wilms tumors and nephroblastoma. Now, that was a mouthful. Um, but those are just some of your oncogenes, your proto-oncogenes and oncogenes. So tumor suppressor genes are genes under normal circumstances suppress growth and check the cell cycle. When their activity is reduced, this results in uncontrolled cell growth. And here we go. So the tumor suppressor genes is like a brake pedal, right? Remove the brake pedal, everybody can do what they want. Your growth factors, signal transduction, nuclear transcription, everything goes what? Haywire. Cell surface proteins, they basically antagonize normal cell growth and development, such as your transforming growth factor beta and the protein in DCC. It's deleted in colorectal cancer. The signaling molecules are NF1, which is, I just talked about that, neurofibromatosis type 1, Adenomatous polyposis coli, APC. Some of these people in familial adenomatous polyposis, by the time they're 15, have over 100 polyps in their colon. Tumor suppressor genes that reside in the nucleus that encode proteins important for the growth and integration and inhibiting signals are your retinoblastoma and your TP53. This is the gatekeeper, very important gene. Retinoblastoma gene, as again, you use the two-hit hypothesis. It uses the CDK inhibitor P21. When it's working, you're not going to have cell growth. When it becomes suppressed, then you're going to have cell growth, and it has to be phosphorylated. So here's our gatekeeper, the P53. Very important guy. If damage is inflicted on a cell, and the cell cannot be successfully repaired, P53 mediates the transcription of genes implicated in the process of programmed cell death. Apoptosis, baby. Apoptosis. You want to destroy those cells. You want your body to destroy those cells. A really good example of apoptosis is when the endometrium sheds for menstruation, when it's not implanted by an embryo. The body says, hey, we don't need this extra tissue. It sloughs off through apoptosis and the woman menstruates. So your normal cell suicide, apoptosis. Mutations in the tumor suppressor gene, P53, are the most common molecular alteration. It causes more than 50% of human tumors harboring mutations in P53. Germline mutations can cause the Li-Fraumeni syndrome, and that causes all kinds of cancers. So if we take a look at apoptosis, it's programmed cell death. You have your normal cell, you have your condensation, fragmentation, and apoptosis. So this is your pycnosis, your karyohexis, and your karyolysis, and then in come the macrophages. Now, events in the process of apoptosis, the stimulus, steroids, radiation, chemotherapy, growth factor deprivation, H3 bitetrahydrobiopterin, maybe. Anti-apoptosis, here's your BCL2, here's your mitochondria, here's your BAX, pro-apoptosis, BAX, this is your cell explodes, basically your BAX, your cytochrome C, apoptosome, nice name. Here's your Cas space 9, Here's your BCLX. Here's your APF BCLX or BAX inactive. When these all come together, basically, we're going to get apoptosis. When this is broken, we're going to get anti-apoptosis, and the broken cells will live. Caspases normally exist in the cytoplasm as inactive zymogens until stimulated through any major pathway triggering apoptosis. So caspases degrade intercellular proteins and activate DNases, which result in DNA fragmentation, laddering, which is characteristic of apoptosis. That's the karyohexis and karyolysis I spoke of. So the mitochondria cytochrome C releases a caspase, causes degradation of the intercellular proteins, so through pycnosis, karyohexis, and karyolysis. It activates the DNases and causes DNA fragmentation, and the cell is phagocytized. Thank you. Uh, and thank you. So a lot to learn, lots of cool stuff coming. I want to thank everyone for watching and see you again soon.